Yeah, I would like to say today's applications of mouse model for FMD vaccine evaluations. So, as you know, the FMD vaccine evaluation is have been conducted in the target animal like pig and cattle. But this method is very expensive. Also, have a time. Take time to long times and laborious. So in this study, we are make a series challenge viruses for the adults, C57 mice. Mainly, we are trying to the O and A and ASHA1 was evaluated in the mice models. That result is the vaccine's evaluation is demonstrates vaccine-mediated protection in the dose-dependent manner. This is obtained the lethal FMD virus, just non-pathogenic virus from the field isolate. Is the inoculated to the C57 mice. And then collect sample from the sera is just the passage in the cell. Passage in the cell second times, and then repeat that process is 5 times. And we got some the pathogenic virus checking, and then we are trying to get growth curve data, the O and A and ASHA1 is a similar data compared to the original virus. And just trying to the pathogenic picture is just before, this is have no the non-pathogenic virus before the passages, but we have some data pathogenic characters O and A and ASHA1. That virus is killed 2 days to 8 days after challenge, depend on some of the virus strains. This is the vaccine evaluations, our own in-house method. So we are own in-house vaccines tested in the models. The survival rate and body weight loss and antibody detection. As compared to the viremia, it's just the ASHA1 is very good result, have no the viremia. So is compared to the survival rate and the body weight loss, also have a good correlation. And the commercial, even though we are trying to the commercial vaccines, just O type is that have some of the viremias. We are trying to the one manisa and the one manisa just the dilute 20 dose, 200 doses. And 3039, we are trying the same method, is just correlate to the viremia and body weight and also espeleza. The another approach is that we have, final. To have some the selection of the adjuvant, so and we want to know the resting protections. So final. We are trying to challenge at the several challenge points in 7 days and 28 days, 84 days after vaccinations. Have some of the data is variations of survival. So, and with using this method, we can protects which vaccine is better in the short time, and the long time, like 3 months, it can do that. Thank you.
Okay, thank you very much. If there's a question from the room, yeah, Tim.
This is taking you back to the sun. I remember it was the first thing I did when I joined Pirbright. We used to check the mice at the weekend. That was Fred Brown's way of putting us in our place, the young scientists. But it's quite interesting, the mouse, you know, there are 2 mouse tests. There's the mouse neutralization test and the mouse protection test. And we were doing a lot of synthetic vaccine studies in the 80s, and we had this problem with very high neutralizing antibody in cattle with peptide vaccines, but no protection. And the single test which actually showed us whether or not the peptide vaccine would work or not was in fact the mouse protection test. So that's what we were doing. That was our index of whether we would get protection in cattle. It seemed to be more meaningful than the cattle challenge tests, in fact.
So, yeah, I know that result, but I think it's just the same condition. So we have to know that quickly to that vaccine is efficiently effect or not. So we are trying to do within the short times, like the 7 or 28 days, we can do that. Which one is better? Okay.
Okay, thank you very much.
Yep.
We have to move on to the last speaker of this session, Claude. You are